All right, so we are all good. So what we are going to do, that you're going to see this slide, and you will see that as we go over, this is probably one of the lectures that, even if you have not revisited the other lectures, if you didn't have to, I will highly suggest that you study for the quiz, everything, no matter what, you will have 2 quizzes due. But when you're preparing for the exam, just go over this recording again, because what we are going to see, that this is what you're going to see. Say first, so all the slides are into here, but I'm not going to stop at every slide. You're going to see that I'm going to stop. I have both of these things so that I just don't have. I'm first stopping in the slide, and there is a star marks in there. So these slides are the one which you want to extra emphasize. Now remember that, don't pick and choose. This is extra important, but if you just know the material from this slide and not knowing the material from the other, it will not cut it. So that's the important part, that at a 4,000, 6,000 level course, you cannot just pick and choose. You need to know it all, but this is extra important for the success in your exam. So that's what it is. So here, what we are talking about in this one particularly, say, just stopping right here. So one of the very important things that we carry the viral genomes as a part of our own material, and we know that a particular type of virus that infected us over the period of time, and you need to know from that. We have discussed it in several slides. Here it does not particularly say on the slide that which is the virus. So what is that virus? Our class of virus, by which you got our which brought these genetic material of the viral genetic material embedded in our genome. The class of virus? Retrovirus, that's correct. Yeah, so retrovirus, that's your answer. So this was one of the quiz questions as well. So these are the type of questions that we are going to ask. So just to know that what it is. So yeah, the retroviruses are responsible for that. That is the only type of virus which can integrate its genome into another genome or us in the host. All right, so after that we are moving into, let's see. So this is a couple of different things that we can just ask. This is also an important slide. So 2 things, you know that there are lots of plant viruses which really doesn't do anything to us. They are just a passive agents, and they are found in the stool. So that's one of the important things, that probably if you just analyze the stool of a patient which is perfectly healthy, doesn't have any disease, and the most likely chances that you will find, if you're just analyzing, it's like I'm just getting a lot of viral sequences in the patient's stool, but the patient is perfectly healthy. If you're analyzing, probably most of those sequences will be of plant origin. So that's also important. And another important thing is that one of the most important one, or we talked about that we just get this virus all the time. This is a virus from the cabbage looper. So remember that. The virus is an insect virus, and it is a baculovirus. So the baculovirus, so it is in your material. So it's an insect virus. Baculovirus is a rod-shaped virus. You can see that it's in yellow itself. And remember that it doesn't infect the cabbage plant. It infects the insect that feeds on the cabbage plant, which is that caterpillar, which is the cabbage looper. So just make sure that you get that. All right. So after that, the very next slide. So definitely know this slide very well, where you can see there is a 3-way symbiosis with the virus along with a plant, a tall grass, and the Carvularia, which is the fungus. So know this as well, and know the association. Remember that the Carvularia thermal tolerance virus infects the fungus, and when the fungus colonizes the grass, Dicanthelium, at that time it offers the heat tolerance or the heat resistance. So remember that if the fungus doesn't colonize first of all, if you just have the grass, and sometimes you can just we can ask some of the like critical thinking questions out of that. If the grass is infected with a fungus and the fungus is not infected with the virus, it's not going to give the thermal tolerance. It's only when the fungus infecting the grass and the Parvularia thermal tolerance virus colonizes or infects the fungus, at that time it only will offer the thermal tolerance. So definitely remember that as well. All right. So then we are stopping at this one, which is the definition of the virus. So definitely remember the proper definition of the virus, what it is. They are ultra microscopic and they can be visualized they can be visualized without the electron microscope. But remember that there are exceptions of the giant viruses like Pandora viruses, which can be visualized by light microscopes. So remember those exceptions. That's very important. So when we say rewrite the definition, just make sure that you also choose the answer which specifically mentions those exceptions as well. So that's it. If I say that it cannot be visualized, just the statement, none of the virus cannot be visualized without an electron microscope, that will be a wrong statement. If there is a statement which says, except that of giant viruses. After that, also just know of virus when we are just talking about the virus. So remember that the viruses, the virus is just basically the name of the virus, just when you are just describing it. It really doesn't tell you the physical entity or the entity in the host or in the environment. We use the word specifically virion. So virion is an infectious virus particle. So definitely remember the definition of the virion. Very important. All right, so after that, we are stopping at this right here. So as I said, that this ties to the definition of the virus. You can see that the 2 micrometer that you are seeing, the pseudo-colored, that image that you are seeing on the top left, those are the typical Pandora virus visualized under the light microscope. So these are the exceptions of the giant viruses which can be visualized under the light microscope. All right. All right, so here we can ask some very specific questions. So you can see that originally the Beijing, they used these candle filters with this ground porcelain. And in the modern tissue culture, we use this 0.2 micron filters, which filter out viruses. So remember that size is very important. Which even cannot filter out the viruses. It will filter out the bacteria. So we do not have, this is the important part, we don't have a filter which really can filter out viruses. So if you are giving a couple of filter sizes, 0.02 micron, 0.4 micron, 0.2 micron, or anything like that, like this, remember that none of them are going to contain the virus. We just cannot manufacture something which can contain the virus. So of course your question will be then, what exactly we do in those conditions. So we prepare those in a sterile condition. Most of the time you just sterilize those. Those are not needed, but I'm saying that the things that you can autoclave those, and you use those autoclave sterile water to make these solutions and all those kind of things. And usually when you manufacture these material in a clean environment, chances of getting a virus in there is very less. But there is no way that if a culture, suppose a material which you are filtering is contaminated with a virus, we do not have, by filtration, we cannot remove that viruses. There are some process, so you go like, isn't there any process? If you just, we are not going to ask. Yes, there could be a process. So what you can do, that on a chromatographic column, you can put some Sephiroth bead added with some antibodies, the immobilized antibodies, which are against the surface antigens of the virus. And if you pour these material through those columns, the viruses will attach, and if you just do the process, you can get rid of it. So there is a process. It's not that it is not possible, but for regular cell culture material, there is no filter. If you are just given a different size just to confuse you, your answer will be there is no filter that can filter the virus for most practical purposes. All right, so let's see. All right, so what are the different ways that we classify the virus? So definitely remember the requirements for that. So just straight nature sequence of the nucleic acid, symmetry of the protein shell or the capsid, whether icosahedral helical, presence absent of the lipid membrane, enveloped or non-enveloped, dimension of the virion and the capsid. This is also some of the ways that we classify. So just straight what is on the PowerPoint. All right. Okay, hold on one second. This is just an extension of the same thing. I do not think that there is any specific thing. We are not going to ask you how we classify kingdom, phylum, class. I just marked this one just as an extension. Just know the virus, how the viruses are classified just with that. But you do not have to remember that if it is an order, you have to tell it viralis. If you have a family, you have to do it viridae. So you do not have to memorize those, not needed. All right, so after that, this is one of the important things that definitely remember the take-home message, that the definition of the virus, and all viruses must make mRNA to be translated by the host ribosome. So remember that if we just ask that which of the following the virus don't have or virus cannot synthesize, your answer will be the translational machinery, which is particularly the ribosome. That is the most important. That turkey-shaped ribosome that cannot be manufactured or made or present in any of the known viruses to date. Otherwise, it will have its own entity. All right, so that's it. So we are moving so pretty fast. Like that, it's very pick and choose. Like you can just imagine that I'm not stopping at every slide. So that's why I'm saying that this will, again, remember that. Don't take the last 2 chapters for your quizzes. This will not work for the quizzes. This will work for the exam. So know everything for the quizzes, though. All right. So second, infectious cycle. So infectious cycle, this is a very important slide. Just know all the definition and in combination that what kind of cell you really need a virus if it wants to infect a cell. You need a cell which is susceptible. That means it contains the virus, and it has to be permissive, which will allow the virus to replicate in there. So other than susceptible and permissive, a virus will not be viable into that cell. So susceptible means that is the process by which, or it contains the receptor for the entering the cell. And permissive means that it can grow inside the cell. Remember that all susceptible cell may not be permissive. A permissive cell cannot be susceptible. So the question is that what is going to happen? Okay, all right, susceptible cell, the virus can enter into the cell, but it cannot replicate in there. So that can happen. It will just be an inert particle just sitting in there, cannot do anything. So that can happen. And the other part is that the permissive, on the other hand, that if it gets inside the cell, it can replicate, but it cannot enter the cell. So here the question is that, okay, all right, so how it is going to enter the cell? This is an artificial process. So sometimes, as I said in the lecture as well, you can package the virus into a lipid nanoparticle, just exactly how your mRNA vaccines are delivered. You have a lipid nanoparticle. This is basically a liposome. You have a coat of phospholipid. Inside that, you have an mRNA in your mRNA vaccine. Here, you put the virus, and you put a phospholipid core in there, mixed with your cell. That phospholipid will dissolve into the plasma membrane, deliver the virus into the cell. So it's a vehicle. Once it gets into the cell by this artificial method, it will replicate in the cell. This is routinely done in viral vectors in the cell culture experiment. We can deliver the virus in a cell which is not a cell which is in the nature get infected, but we can have some permissive cells which doesn't have the receptor but has the ability to replicate the virus. We can do that. So very important slide. All right. So, all right, cultivation of the cell line. So just remember that what kind of cell line that we use to propagate or do the experiment in the viruses. In the laboratory, we can use any type of cell line to study the viruses. But the most important, remember that that many of the cell line, which are continuous cell line, that means they will live forever if you keep passaging them. Those are mostly aneuploid, and as I said, they are usually, these transformed continuous cell line are usually derived from a cancerous tissue, almost always. And that's the reason that they have this aneuploidy, aberrant chromosomes, and may have oncogenic DNAs in there. So if you are making a vaccine, so suppose you are making a vaccine and you went to FDA, it was like, I have a very good vaccine candidate and I'm just growing this one, this is the approval. So the FDA will ask you that where you are manufacturing your vaccine. You tell that I'm just doing that in this human epithelial cells like in HeLa. It was like, okay, all right, no, please go back. Because you have this aneuploid cell line and your virus may be contaminated with the oncogenic DNA, and it's absolutely no. You either have to use a primary human cell line, which is not oncogenic with a diploid DNA, or some specific type of cell like V38, which is also diploid cell strain. You cannot use a continuous aneuploid cell line to manufacture DNA. So you can have some critical thinking question. Remember that many of the questions, as I said, that you will have a paragraph to read. So you will have a story out there, just as I said, this FDA story. You will have something like that, a paragraph, and you just answer that questions. So, all right. So after that, the cytopathic effects. So know what are the cytopathic effects. So here, mostly what we are looking at is the rounding of the cells, and they are being detached from the Petri plate or the tissue culture flask. That's what we are seeing. The next one, what we are seeing is some giant formation of the syncytia. Say, for example, if we just asking like this, that if you are culturing a virus in the laboratory, and you just got a virus, new strain, you just got it from somewhere, and you just put it in a cell line, and after that, what you are observing, that you are observing giant cells with multiple nucleus, just exactly what you are seeing. So at that time, if you just ask that what it will be your conclusion will be that virus is causing this syncytia formation. Syncytia is plural, syncytium, singular. Syncytia formation, which is basically fusion of multiple cells to make these huge, large viral factories. So that will be your observation. So after that, some of the important ones, that I specifically arrow marked those. So that's why it is that if we just ask that which of the following are cytopathic effects. So just remember that this is a nuclear shrinking, proliferation of the membranes. These are very important. Then syncytium formation, we just saw that, very important. Rounding and detachment of the cultured cell, we both saw those two in the middle. And the fourth one, which is very important, is the presence of dark inclusion bodies. Remember that these inclusion bodies stain very dark, either by negative staining or even light microscopic staining. Remember that the inclusion bodies can be seen in light microscope, like the Negri bodies, which is produced by the rabies virus. You can see under the light microscope. You are not seeing the virus, but you are seeing this inclusion, which are the viral factories where the viral proteins are being made. And as they are very protein rich, so this is just an overview of understanding, putting that thing, that why it should appear dark. So usually what we do, that many of these stains, including some of the stains that we use in electron microscopy, if they have an affinity for a negatively charged groups, which the proteins have lots of negative charges on them. So if the dye is positively charged, there are many of them, they will go and bind in there, and they will be appearing as dark inclusion bodies. So just know these. That's important. All right. All right. After that, we are looking at the plaque assay. So definitely remember that the principle of the plaque assay. So basically, what happened that I'm just saying that an infectious virus particle, when allowed to infect a monolayer of the cells, and the viral dispersion is restricted by an overlaying gel like agar, the virus kills cells around the cell, that it initially infected, which cause a clear zone due to death of the cells, and this is called a plaque. So that's your definition of a plaque assay. And the second question that we sometimes ask is that why after infecting the virus, we take a plate, we infect it with the virus, just allow it to adsorb. Remember that the word we use is adsorb, not absorb. Adsorb is just when the virus is sticking onto the surface of the cell, probably to this receptor. Then we take the excess virus out so that too much viruses are not there, just the ones that infects just one cell. Take out that and then overlay the cells with an agar or CM cellulose, no matter what. It should be a gel-like material. And we ask that what is the function of that gel-like material? Remember that what happens that when the virus is infecting these cells in the middle, say for the first one, the cell will die and may float out, and these viruses which are coming out, easily coming out into the media, if it's liquid, and diffuse around. If you have the agar overlay, this movement is restricted. So the viruses which are just next cell, they can just go in there, but they cannot move. Say for an example, if I just pointing out, if the viruses which are produced here can go in these cells in the vicinity, but they will not be able to infect in there. They will gradually do that. So that is the purpose of the agar, semi-solid media, or CM cellulose. Just remember that. All right. So after that, we are looking at this. You do not have to do any calculation as such, but just remember that when we calculate the plaque, what is the outcome, how we express that. And remember that the number we count, and with the dilution, we call this as a plaque forming unit, or PFU. This is how we measure this is the quantitative measuring unit after a plaque assay. So the plaque assay, what do you measure? You measure the amount of the viruses which are viable, means that is active and infecting the cells, and you just express them in PFU, or plaque forming unit. All right. So that's the same thing, the same thing, and nothing else. Do not have to do the clonal thing, but just remember the PFU or the plaque forming unit. That's good enough. All right. So here also same exactly. What is the definition of the particle to PFU ratio? Number of physical particles over the number of infectious particles. That is your particle to PFU ratio. Now here, if I just ask that what you mean by the particle, like particle, of course virus particle, but what really is meant by the particle? Remember that if you're just giving a test tube and it's like, okay, all right, there is 10 million viral particle in there, so that's fine. So, but what you mean by that out of this 10 million that is in there in the test tube, there are many of them which are defective. It is just the viral particle, but they do not have the ability to infect. The ability to infect is only the virions which are the infectious one, and that's what it is. So out of 10 million, 5 million of them infectious, so those are the infectious particles. So 50% of them are viable. So remember that. When we mean by the particle, it means that it contains the infectious and non-infectious particle. Remember that. Again, I'm repeating. When you are seeing that the word in a particle to PFU ratio, here if we just ask that what do you mean by the particle, remember that the particle means the particle which are infectious as well as non-infectious. So that's the important point. So that's why there are 10 million, and half of them are infectious, half of them are non-infectious. Then you have 5 million infectious, 5 million non-infectious. That's basically what it is. So just the concept. All right. So here also we are looking at, this is also, we talked about several times, and now you know in the later chapters the principle of that as well. So know the principle of the hemagglutination assay very well. So basically some viruses can clump or agglutinate is the word, the red blood cells to form a lattice that don't sink at the bottom of the assay plate. and will give a distinct appearance, difference in appearance from the non-infected red blood cells, which without the hemagglutinin sink at the bottom of the well to form a red button. So all the ones which with the red button, these are the ones which are without the virus. The red blood cells are sinking in the bottom. And again, remember that here we explained that why you are seeing a 1:4 a red button. These are your perfect experimental. These are the lattice where the red blood cell is not sinking in the bottom. That's why you are getting this lattice. Now what you have, that you are started to seeing the viruses are too dilute, and they don't have the ability to clump or form the lattice or form the clump of the red blood cells. That's why they are tumbling at the bottom. So this is probably the one which is the correct one. Now you can see that when there is a 1:4, when there is a high concentration of the virus, it also sinks to the bottom. So you now know that the virus contains the neuraminidase on its surface, and it's not many, but there are some. But in a high concentration, it becomes quite a bit, and that will basically cleave these sialic acids, which are responsible for clumping them together. And that's how the very high concentration of the virus will do that. So that you know from the previous one. All right, so after that, know the basic principle of the ELISA or the enzyme-linked assay. So we can just ask, is that something like that, which of the following assays involve the principle of immobilizing a viral antigen or a viral antibody on a solid support, assaying the interaction, binding or not, which can be determined by a secondary antibody and an indicator. If we just give the definition, you just know that it is, we are talking about ELISA or the enzyme-linked assay. All right. So here, very important idea. So definitely remember this. So single or multi-step growth cycle. Now, we asked some of the questions in the quiz that you have infected these cells for a multi-step growth curve or a single cell growth curve, and we asked some of these numbers, like a 10, 100, 10.1, 0.01, something like that. You have to choose that. So remember that usually if we are trying to do a single step growth curve, usually you use a high MOI. And what will be that magic number? It's 10 or above. Usually that's the number, 10 or above, if you see in that. Single step growth curve, 10 or above. If it's a multi-step growth curve, 1 or below. That's the thing. You do not have to do any calculation. And I can just quickly go back and say that where we get these numbers. So you get it from the Poisson distribution, but you do not have to do any math. I'm not going to ask you math questions here. But this is the thing that what we are trying to see. So if you look at the 10 to the power 6 cells infected with 10 MOI, you can see 45 cells are uninfected, 450 cells received. So 99% of the cells are infected at the same time. You will get a single step growth curve. But if you look at the 10 to the power 6 cells at an MOI of a 1 or 0.001, in one case, 37% of the cells are infected, whereas 99% of the cells are not infected. So what's going to happen in the middle 1 or 0.001? Some of the cells are infected, virus is coming out, infecting some other. So, and then other. So for the first burst, you get a step. Then the next burst, you get another step. That's how you get a multiple step growth curve. So you can be given just a growth curve, just how it is. And we can just ask, right here, that we can just give you a curve like that. And, or we can just give you that, okay, all right, we have infected the cells with these MOIs, like say 110, 1,000, 1.01, something like that, and which will be the growth curve that will fit in there. So remember that anything 10 and above will be a single step growth curve. Anything 1 or below will be a multi-step growth curve. So that's important. So, all right. So that's exactly what we are talking about right here. So nothing else, we don't have to do any math issues with that. All right, so right here is the experiment by the Hershey and Chase, and just know the principle of the experiment. So basically what they did, they labeled the viral protein with the radioactive sulfur. Remember that, the radioactive sulfur and the viral nucleic acid. So remember that radioactive sulfur is a part of the protein. Remember that. So methionine is where, so they can just use the methionine, which can just go in the that, or cysteine if it is present in the that also contains the sulfur. So the 35S is usually for protein labeling, and then you just use the alpha 32P as well, phosphorus, to label the nucleic acid. And the presence of these radioactive molecule in the proteins and the nucleic acid on the progeny bacteriophages. So they found that there is presence of the radioactivity in the genome, basically, because it's a bacteriophage DNA genome. So the progeny phage do have one strand of the parental DNA after replication. So that's how you know exactly that that was the genetic material. But the proteins were all synthesized new, so there was no radioactivity in the protein in there. So know the basically the principle of the experiment. That should be good. All right. So after that, you have done this many times. So just remember that how you are trying to do this this process as well. So if we're just looking at double-stranded DNA, so suppose you have this sequence. This we call as a coding strand. Also, sometimes we call this one as a sense strand. Remember that. We call this a sense strand. On the other hand, this template strand is sometimes called also antisense strands. Another name, we call this, each strand could be the complementary to the other. So the 3', 5' strand, if we just look at the top with the template strand, so the coding strand is complementary to the template strand, and the other is true as well. The template strand is complementary to the coding strand. So just based on that, you just try to find out that how you can make the viral mRNA. So say for an example, this is the viral mRNA, and how we actually are making the mRNA. Remember that this is when it is in the DNA, we call this as a triplet. So GAC-TCG to a triplet. When it is in with the EU, we call them codons, which is each codon is basically for one amino acids, or codes for one amino acid. So triplets or codons are basically exactly the same, except the codon is the thymine or the T's are replaced by an U. That's the difference. So how you reach into the viral mRNA? So let's start with the double-stranded DNA. Remember that there is always a shortcut that you can do, but that's not the best way to do it. So if you just carefully look at that, if you see that GAC, TCG, AGC, this is the code in here. And if you just replace that GAC, like GAC, here T, I just replaced UCG, AGC is exactly the same code. But always try to remember that how you reached or how it is manufactured in the cell, that is important, very important to do that. So what will happen, the RNA polymerase will sit on that and make a bubble in here. And it will move from the 3' end to this. And while doing this, what it is doing basically, you can easily see that right here, you have the CTG, and it will just put the complementary strand. Basically, it's reading this and putting the complementary strand, and it is moving from 5' to 3' direction, making this. Remember that all the polymerases move from 5' to 3' directions, while reading the 3' to 5', which is the complementary strand. Again, remember that DNA, RNA polymerase, they all move from 5' to 3', reading the 3' to 5'. As it is reading, this is what it is doing. So it is C, it is putting a complementary of that, which is G, of course, and you are putting a G. Then it's reading that T, it's supposed to put, as it is an RNA polymerase, it's not going to put an A, but it's going to put, of course, it's going to put an A. But so this is exactly the same. but so you have GAC. But here, see that as soon as you got an A, the RNA polymerase cannot put a T, so it puts an U here, and the CG is the same. So just remember that if the RNA polymerase is reading this and making a complementary of each of these with an U if an T appears. So that's how it is. So let's take a look at that there are some of the plus strand RNA viruses. So if we just ask that, suppose this is exactly the full genome of the virus. So just it's written as viral mRNA. And here I'm just taking this viral mRNA out and telling that, okay, all right, this is not the viral mRNA. I give you a 5' a sequence like this, 3', and this is the sequence, and saying that this is the viral genome. This is a complete viral genome, just a hypothetical case. So to make a mRNA, what it needs to make. So remember that if it is trying to make an mRNA from here, it cannot make an mRNA from here because this is an mRNA by itself. It can be read by the ribosome, but it cannot make an mRNA. So what it will have to do, remember that it has to make a negative strand right here with its GC, AU, CG, and then UA, CG, GC. So you have to make an intermediate negative strand. On this negative strand, you get that mRNA back. A plus strand and these are both are the same, but you need to first manufacture this strand and go back. On the other hand, if we just ask you this, that you have a negative strand RNA like 3' like this, CUG-AGC. So 3' it starts with this. If you just given like this is a full genome sequence, okay, negative strand RNA virus. So you can see that for this, if I just ask for the negative strand RNA virus, if that is the genome, what intermediate strand it needs to synthesize to make its mRNA? What will be your answer? Again, repeating. So you have the negative strand RNA. I'm saying that is the genome of the negative virus. So what will be the intermediate strand? Does it need to make an intermediate strand? Everybody good with that? It just uses that negative strand template to get the mRNA. You do not need any intermediate. So remember that for your negative strand, you do not need a template. You just read the RNA polymerase, RNA-dependent RNA polymerase will just read this and make that mRNA. But for a plus strand, you need to make that intermediate and get back to that. So just definitely spend some good time to practicing that. And just the typical question that you had in your quiz. So just practice a little bit on that. So you can get multiple questions from here as well. All right. So then we are going into the Baltimore scheme. So definitely remember that the basic question that we ask, that why we have placed the mRNA in the middle of that scheme. So remember that the main thing that why we put that every virus must make mRNA. Before, like, of course, it has to make its genome, but that is the key, that it has to make the mRNA to make the different proteins, including its enzymes as well, before it can do anything. So that's the reason that it has been put in the middle. So that's very important as well. So after that, we are looking at some specific questions. Let's see. So just remember that what are the different things that can be attached to a DNA genome. So remember that with the genome, sometimes you can have an covalently attached proteins as well. So remember that is one of the important one. That is sometimes you like genome, why there is a protein. There are some of the genomes with the attached protein. So I have not put it in there. I'm just going to show it in somewhere else. You're going to see that in a second. Just give me 1 second. All right. So right here, we can see that right here, what we are looking at, that the DNA viruses, these are double-stranded DNA genome. So if we just ask that once the DNA virus goes into the cell, which of the following is basically needed to replicate the DNA? So remember that, as we have said, on the left-hand side, genomes copied by the host DNA polymerase. So it can use the host DNA, our DNA polymerase, to do that. On the other hand, for some larger viruses as well, genomes encode the DNA polymerase. So both are correct. So remember that not only the viruses use the host DNA polymerase. If it is a double-stranded DNA, they can use a host DNA, of course, DNA polymerase in some of the cases, but there are some viruses which code their own DNA polymerase. So remember that both are true here. So that's important. Also, after that, so this is a double-stranded RNA genome. Remember that double-stranded RNA genomes, say for an example, in the Virology family, they can be segmented. That means that the genome is made up of just chromosome-like multiple segments. So you have this, you can see that large, medium, so you have 3 large, 3 medium, and then 4 small segments. This is what we find in a real virus. So double-stranded RNA genomes can be segmented. So that's important. So you have 10 double-stranded RNA segments. Here is that right here, what we are talking about here, the example of this where you have a protein attached to that. That's why I just did not stop at that. So you can easily see that example, that what are the different things that can happen. So here, sometimes a protein can be attached to a viral genome. That's what you basically get. Here, from this one, we are getting then some double-stranded RNA genomes can be segmented. That's also important. All right. All right. So here also, we can ask the question the other way. See, remember that this is revisiting that question. If you have a negative sense RNA genome, you can use this directly as a template for the mRNA, which can make the protein. But how will you, if we just ask the question other way, that how will you make a minus sense RNA, which is genome, or what intermediate need to make? When it needs to make its own genome, remember that at that time, it needs a plus RNA, which is the intermediate, and on this template, it can make the minus RNA back. So remember this process as well. So you can ask, is like, so basically the mRNA that is being made, it is the same thing in here. In some of the cases, it could be the same thing. In some of the cases, it cannot be. That we were not going to ask, but I'm just for the clarification purpose, I'm just trying to draw. So suppose you have a genome like this, which is your 3' and a 5', and this is your negative sense genome right here. What you can have, that you can have different segments, and this is say 1, 2, 3, and 4. There are 4 genes in here. So what happens that when you are making an mRNA, the mRNAs are made like this. You have an mRNA from here, you have an mRNA in here, mRNA from here, mRNA here. See that they are different lengths. But these will be like this. But when you are going into the route of making the genome RNA, which is, let's change the color, genome RNA, you have this same template. You are going to make a full length, which is plus 5', 3', and it contains all the segments 1, 2, 3, 4 in reverse. Remember that, it's in reverse. So you have the plus, this will be your intermediate. This is your intermediate RNA. On top of that, I'm just again going to see that the color, let's change the color to olive now. So it's going to make the full length of the 3' to 5' right here, and this is your minus sense. So this will have basically the gene 1, gene 2, gene 3, gene 4. So that is the difference in between. So basically, this is this. So that's basically the difference in between them. Definitely remember that. So what is the difference between the mRNA? Basically, technically they are the same, but the genome is full length, whereas the mRNAs many times could be subgenomic, which are smaller. All right, so let's take a look at this. So after that, this is also another important one, that remember that there could be extensive, just the fact that we just always draw the genomes as a linear genome, but it never exists like this. There could be really very complex secondary and tertiary structure and long distance interaction in the RNAs as well. So definitely remember that, and that's how it is. So here you can just see an example of a picornavirus with a protein attached. We say that in somewhere else as well, but if you just look at its genome, it is this complex. So here is a question. So how do you think that the picornavirus was named? Can you think about that? The name something tells you something. Anybody can imagine? Just for fun. That why it is picornavirus, what the scientists were thinking while naming it. Yeah? Say that one more time. The spike protein is like a crown on the surface of the virus. Well, it's so for the picornaviridae family, remember that it's a poliovirus. It's not enveloped though; those are naked viruses. But it's just the size. They are extremely small, 30 nanometer. So it's pico, and it's an RNA genome. So picoRNA. So that was the thought process in there. All right. So let's go to the next one that will do it for this chapter. All right, so very important slide in the structure of metastability. So here, just remember that what we mean by the viruses are unstable. So pretty much everything that you need to know here, that the function for that metastability is for that they must protect the genome, and they must also come apart to release the genome into the cell. And metastability means that they have not obtained the minimum free energy conformation. And think about that this is what we call as a spring-loaded. So we're talking about this herpes virus sometimes, and just like the bacteriophage, even it's kind of a really amazing, like I just probably just for the lack of time, it is in the blurb, but probably I have not said in the class, that the herpes virus genome is huge. And it is put with approximately 700 psi pressure into the viral particle. So think about that how in that tiny particle, 200 nanometer wide, can put something with 700 psi pressure. So what happens that once the virus basically lodges the herpes virus goes and uncoats into the endosome, after that, it is carried all the way into the nucleus. It sits on a nuclear pore, and that causes the information of that portal or the Stargate to open. This is not needed here, but I'm saying just as soon as it's opened, just the sheer pressure delivers that DNA or inject that DNA into the nucleus. So it's really amazing that how it is done. That is one of the information that is what we call as a spring-loaded. Very classic example. That example is not needed, but just for your long-term memory. So then the next one is very important right here. So remember, the subunit is a single polypeptide chain, or the protein, a single protein is a subunit. On the other hand, remember the structural unit is the protomer, or it's also called the asymmetric unit. And this is the unit from which the capsids or the nucleocapsids are built. So that is very important. So the definitions are important. So this is an important slide as far as a definition goes. All right, so after that, we are looking at this symmetry rules. So definitely remember that which of the following describes the virus symmetry for self-assembly. Remember that each subunit has different bonding contacts with its neighbor, and self-assembly of the virus particles like there is always the bonding contacts in the subunits are usually non-covalent. That's basically your answer. So remember that. Sorry, take it back. So what I said before that was wrong. So here, this is the important thing that we just can. Give me 1 second. Right here, so there is a lesser pointer. That's it. So this is the most important part that you can underline. So if we just ask as that which of the following describes the symmetry of the self-assembly, so remember that each subunit has an identical bonding contact with its neighbor. So if I just change this word to each subunit has a different bonding contacts with a neighbor, that will be a wrong choice. Remember that. If you just make it a different bonding contact, it's an identical bonding contact with its neighbor. And these bonding contacts are usually non-covalent. And what once we mean by non-covalent means that these bonding contacts could be hydrophobic protein-protein interaction. These could be hydrogen bonding. That's mainly the most one of them are hydrogen bonding, and also van der Waals force. Sometimes it could be electrostatic as well, but that is secondary usually. So mainly hydrophobic interaction, hydrogen bonding, and van der Waals force. These are the ones which are, most of them are weaker forces, but they are non-covalent. That can be reversible. A covalent bonding, you have to digest it or chemically split that bond. You cannot reverse that process. So that's why you cannot covalently attach that, basically. So that's the important one. And again, remember that although the rule 1, each subunit has an identical bonding contact with its neighbor, remember that for all practical purposes, that is true in here. But when we have the quasi-equivalence theory in larger and the larger virus particles, we actually violate that theory as well. So that's another important thing. All right. All right, so here we are looking at the icosahedral symmetry. And remember that an icosahedron is basically with 20 triangular faces. Each is an equilateral triangle. And so here is an interesting thing that if you just really think about that, why you have to have this symmetry to build a particle. I'm just trying to see is like, have you thought about it that why you exactly need this basically? If I just trying to do this structure, I'm just trying to say right here is not exactly, well, hold on. 5, 6, okay, all right. So basically, if I'm just drawing this is basically a hexagon, okay? So you have 1, 2, 3, 4, 5, and 6. Now, if you have to make a structure which is enclosed like this, if you try to make it with an hexagon, you will not be able to actually do that. Because what happens is that this is a planar structure. So to do this, basically what you have to do, that you have to delete one of this. If you have delete one of this, it becomes a pentagon like this, and this will fold like a cap. If I'm looking at from the side, this will fold like a cap. This will fold like a cap, and of course from the side. So you can enclose a structure. And that's the reason that we have these 5-fold symmetries coming into the icosahedron, because you need this folded structure to do that. But again, as the structure grows bigger and bigger, you have the 6-fold symmetry as well. But here, these are some of the basic things that you need to know, that What are the different properties of this? That it produces a solid 20 faces, each equilateral triangle, allows the formation of a closed shell with 60 identical subunits. Remember that you need for this, you need a 60 identical subunit. You have a 3-fold, 5-fold, and a 2-fold axis. And the T number, which we are going to see, describes the number of facets in a number of facets, basically per surface, basically per triangular surface. So what we are looking at here, say this is your T equal to 1, and if you just look at that, this is built with just one protein. You have 1, 2, and 3. Here, all these proteins are the same protein, so all the subunits are the same. So basically, in this is a trimer. So you have a one monomer here, one monomer here, one monomer here. You have a trimer. And then things get complicated as we just move over that. So here, what we can just ask from these questions from here, that if we just ask that, say for example, if we just increase the T numbers accordingly, once we are just going over and over that T number, say for example, T equal to 2, 3, 4, 5 as such, you can see that along with a 5-fold axis, a 6-fold axis also generates. And that's a problem. So that's a problem, means that now the concept that quasi-equivalent comes in, that there are two structural environments, the two proteins in two different places are going to bond a little bit different. And sometimes it is very difficult just to understand that how it is happening. So for an example, see that there are 3 protein hydrogen bonding like this. One here, one here, one here. And just think about that just to accommodate those 6 in a different area where you have a 6-fold symmetry, these 2 contacts remain here. This is distorted a little bit, and you have a bonding contact here, and like this goes away. So this goes away, and you don't have this contact anymore. You have an extra contact here. So it's hydrogen bonding with a neighboring amino acid, and it looks the same, but just allows the protein to accommodate in that shape. That's basically what we mean by the quasi-equivalence. So for an example, now we just can ask also another questions from here, that which will be the following number of subunit, if found in a virus, will not form a quasi-equivalence. So what will be your answer? Say 60, 120, 240, 720, if I just give these number. So these are the number of subunit, and out of those subunit, which one of them the proteins will not form quasi-equivalence? Again, 60, 120, 240, 740, 720. 60, remember that, 60 will not form the quasi-equivalence. It's made up of one protein, that's it. So because if T equal to 1, it does not have a 6-fold symmetry. So it has only 5, 3, and 2. So as soon as a 6-fold symmetry comes in, that quasi-equivalence kick in. Before that, if you do not have that 6-fold, which you do not have in a T equal to 1, you will not have the quasi-equivalence. So 60 subunit, it will not form. So that's your take-home message. That should be good enough. All right. And also remember that say for example, when you need the T equal to 1, that how many of the proteins that already I said that how many proteins you have in per triangular face. So remember that you have a trimer, 3 proteins will be your answer. Yeah, that will do it. All right, so here also you can be given a very simple math formula that you can just, you do not have to use any remember that during the exam, just keep the cell phone somewhere back in your room, just don't put on your desk, because if you just need to call me for some reason, you can reach, but not on your desk. You do not need a calculator as such to take the test. You can have a very simple arithmetic problem or something like that. Say, for example, if I just say that you have your H direction, the vector is 5, say for an example. Let's see. So here you have a number, but if I just change this number to say a 5 or 2 or something like that, and how can you calculate the number of subunits and basically the T number? So that's basically just plug in, just all you have to remember for that process, that T equal to H square plus HK plus K square. So if I just give you say 5 and 2, so if I just give you a different number than this, so I know it's redundant, but I'm just giving 5. This is H equal to 5, and then K equal to 2, if I give this number. So if it is 5, then this becomes 25. Everybody get good? And then plus this one is how much? HK, 10. That's correct. Plus 4. Everybody good? So 39 will be your T number, and what will be the number of subunit? You multiply with 60. What will be the number? 2340? Did I pass or fail? Good? Yeah. So just multiply. I mean, like, you should be able to multiply these small numbers, hopefully. So that's basically what it is. That you have to calculate. So this is, it will have almost an identical problem. That is the most that you can get. Yeah, so that's should be. All right, so let's see right here. Yeah, so that's all good. So here also another thing is what happens is, so if I just ask is like here, the triangulation number is T3, and there you have the VP1, 2, and 3. These are buried, the VP4 is buried inside the capsid, remember that. And what we are going to ask that if this is what it is, what kind of interaction among these proteins are expected and observed at the 6-fold axis between the VP2, VP3, in contrast with the interaction of a VP1 at the 5-fold axis as per the Kaspar-Klug's theory of quasi-equivalence. So again, I'm repeating the question. So suppose this is, I just described this, that you have a VP1, VP2, and a VP3, and asking, suppose at 6-fold symmetry, here you have a 6-fold symmetry, here you have a 5-fold symmetry, and we're just asking that what kind of interaction that you are just going to have. Just remember that as per the quasi-equivalence theory, the bonding interaction between the VP2 and the VP3 at the axis should be similar to the interaction between the VP1 subunits at the 5-fold axis, but in reality, they are very different and does not conform strictly to the principle of the quasi-equivalence. And again, I'm just saying that the interaction at the 5-fold axis and the 6-fold axis do not have the identical interaction as per the quasi-equivalence. Technically, so that's basically what it is. So again, just don't confuse the questions that has a, so you have VP1, VP2, VP3, and do not get confused that I have put all these names, what it is. I'm just asking that you have protein 1, 2, 3, protein 2, 3, 4 or something at 5-fold and 6-fold. What will be the type of protein of interaction? You're just asking that due to the quasi-equivalence, these interactions are not identical. That's your take-home message. If I put a story on top of that, don't get distracted with that. So that's basically what it is. All right. That will do it. So here, that was the thing that what we were talking about, that this is the herpes virus, this target, not needed, but I'm just saying that it just packs it with such a high pressure that it delivers the DNA like that. Okay, so let's see. All right, so here also we can just ask some of the questions about the adenovirus, that some of the fill in the blanks type of questions, that where the CAR binds. Remember that the CAR binding site is, so the CAR binding site is on this trimer, on this fibers, remember that. And on the other hand, the integrin actually binds on the base. So just remember this. This is information type of questions, basically. So the CAR basically binds to the knob region, basically, and the integrin binds to the penton base. That's basically your question. All right, so here also the sialic acid, remember that the avian influenza and the human strains are different. You have a 2,6 and a 2,3. Basically, so if we just can ask that, what is the significance for this? The significance is that modulates the host specificity. If you have a 2,3 linkage, as you can see that if you have a 2,3 linkage, it is easier for the virus to infect the avian strains. If it is a 2,6 linkage, it is easier for the virus to infect the human strains. So that's basically, so it modulates the host specificity. All right, so after that, if we just ask that the sialic acid, which is the binding portion for the influenza virus hemagglutinin, where does it bind? So remember that this is the extra ectodomain or the extracellular domain of the HA protein or the H protein, and this is the globular head. And you are looking at the globular head from the top. If you are looking top down, this is the middle of the globular head, and here sialic acid bounds. So if you just ask where it binds, remember that it just binds at the very top at the globular head. And that is obvious. Remember that all these spike proteins, the binding surface is at the very top. Some of the places it could be a canyon, non-enveloped viruses though, but here it is at the very top. All right. All right, so here HIV here, so remember that the basic mechanism of that, we can just ask like this, that which of the following statement for the HIV fusion is basically correct. So if we just say that remember that right here, the cellular CD4 interacts with the viral SEU, which is the GP120 protein, which changes the conformation and interact with the other co-receptor, which is the CCR5 or CXCR4, any one of them, CCR5 or CXCR, this is written as CCR, but if you just read your material, you will see that it could be either CCR5 or CXCR. So upon this conformation change, it's bind to this, and this induces the conformational change, this interaction, to this transmembrane domain, which flips and inserts its fusion peptide into the membrane. And that's basically the process. So again, repeating, CD4 changes the conformation of the SU. SU conformation change allows it to bind with the co-receptor CCR5 or CXCR4, which induces the conformation change of the TM, where the fusion peptide flips out and inserts into the membrane, and then leading to, you have seen the hairpinning happens, and the top membrane and this membrane will basically fuse. All right. Right here. So here, one of the most important thing that for flipping the fusion peptide, this is in red. What is the important thing that is happening? Remember that it is the proton. Acidification of the endosome is the most important part. The endosome is acidified, and then these protons are again pumped inside that allows the fusion of the membrane. So acidification of the endosome is very important. All right, so after that, know the mechanism of the, oops, sorry, I do not need that. The Ebola virus, remember that the Ebola virus, that what exactly happened, that the Ebola virus basically binds to its receptor, and it just goes into the endosome, where at the acidic pH, a portion of this glycoprotein is cleaved, the glycan cap and the mucin keep is cleaved, and it can interact with the NPC1. Remember that this happens in the endosomal pH, which is acidic. It does not have this at the neutral pH. Remember that anything that happens outside at the plasma membrane, that is at a neutral pH, and that will not be correct. So remember that it will not be correct at the neutral pH. It happens in the acidic pH. So because in the endosome, all the proteases which are responsible for there, this happens in the neutral, in the acidic pH. All right, so just to know the mechanism of that. All right, so here also another question, that what is the mechanism? So here we can have a question, see that right here in the canyon where the CD155, the receptor binds, underneath this canyon, there is a pocket. And if you just carefully look at this pocket, it is very hydrophobic. And see that these are the aromatic rings. It's a hydrophobic compound. So you can just think about that you just suppose that we are just asking a question that you have developed a drug which is a hydrophobic, and you are just putting that drug in that place, basically. And what will basically happen? So that basically, if the drug sits in there and doesn't leave, it will not allow the viral infection, because it will not allow the unplugging. So only when this hydrophobic compound leaves that will cause the VP1 and the VP4 to flip out, and this VP3 plug moves away. So it has to be coming out from that canyon. So that's the important part for this. So cell lipid, and remember that this is a hydrophobic pocket. That's your hydrophobic, so that's your important part. So if you look at that, it's very hydrophobic. So definitely underline that word, hydrophobic. Then here, the uncoating of the reovirus, that how reovirus basically uncoats. Remember that it goes into the endosome. Endosomal proteases digest the outer core, and the inner core proteins are hydrophobic. That basically binds onto the membrane. Remember that outside, the outer core is not hydrophobic, because it will not be stable if it is hydrophobic. Only once the outer coat is digested in the endosome, the hydrophobic proteins of the inner core is exposed, which basically binds with the membrane. And that will do it. Good timing. So it's very comprehensive, but again, remember that don't use this for the quiz this weekend. Use this after the quiz. So along with that, also know all your quiz questions as well. You may not find the quiz questions word by word. They could be a little bit different, but knowing all the quiz questions will help. And sometimes after 3 attempts, you should get pretty much all of them. If you just do not, you can just compare the question banks with each other. If you see that everybody is getting all of them, that will help each other as well. So 50 questions, 75 minutes. All right, we will discuss the honors things. That will be easy. That is just for, that is popular science on SARS. So that will not be difficult at all. All right, folks, have a good weekend. So invest some time. You will not regret.